Most patients who first receive chemotherapy, they end up performing pretty well. They have a complete response and the tumor almost completely goes away. It's clinically undetectable. And then at some point, whether it's 6 months or 6 years later, that cancer comes back to form this recurrence. And this tends to be highly resistant, and there's not so many therapies or targeted therapies for the recurrence either. So we have to assume that the tumor never fully went away if it's coming back after that time. And this means that, it really points to the inadequacies of chemotherapy in general in the clinic, that it can't fully target the entire part of that tumor. And there's always going to be some resistant cells left behind that then go on to form this recurrence and is leading to this lethality of ovarian cancer in general.
Ovarian cancer itself is typically detected fairly late in its development. That's also why the prognosis of the patients is so low. But as James just mentioned, initially chemotherapies show, or the tumor show a high response rate to the initial chemotherapy. However, there seems to be a small subset of tumor cells that are resistant, that remain, and these are typically undetectable. But then after a certain time, these tumor cells grow again and form a new tumor that then is actually resistant to chemotherapy.
Well, it's really just trying to find these parts of the tumors that initially don't respond to chemotherapy, to understand which part of the tumor has resisted and gone on to form the recurrence. Recurrence being the most deadly part of ovarian cancer. We need to understand which parts of the tumor go on to form it, and then how we can use that information to potentially identify new treatments for recurrence. And that's, I guess, where our project comes in. So we're using essentially imaging technology to look into these tumors in a very high amount of detail and to understand which particular regions of that tumor and which parts of that tumor are the parts that can resist chemotherapy and form that recurrence. So we use a technology called imaging mass cytometry that Bernd previously developed, and we're using it to identify huge amounts of information in these tumors, such as resistance pathways to chemotherapy, such as immune infiltration, vascularization, and all of these essentially aspects of a tumor that could potentially lead to that tumor resisting. And we can look at this in hundreds of patients, and across all of those patients, see if particular spatial trends are associated with outcome to a particular therapy. So in doing so, we have a lot of strength to essentially predict whether a patient will survive, whether they're going to respond to chemo, and how we might be able to tailor a new treatment towards them.
An important next step will be that with this imaging method, we can also detect which potential drug targets are present in these tumor cells that actually resist chemotherapy, or whether there might be drugs available that overcome these resistant mechanisms. Then these cells again would respond to chemotherapy. And so we can gain in-depth insight into the different types of chemoresistance of these patients, and hopefully also identify what might be the best combinatorial treatment that one could provide to those women to then make these tumors either chemoresistant again, or use a different drug that then also will show high efficacy against those cells. I guess in general, in cancer, a major, if not the major hurdle, not only for research but also for the clinical setting, is that it's an incredibly heterogeneous disease. And that means that the same clinical subtype of cancer, in this case ovarian cancer, if one uses, for example, our technology to comprehensively determine what is present in this tumor, which type of cells, what they do, then you will see from patient to patient to patient actually big differences. And even within a single patient, you will find that there are different types of tumor cells present. And that also is an explanation why many tumor cells respond to chemotherapy, but others will actually resist. And this enormous heterogeneity, these enormous differences within a patient in the tumor, but also then across patients within the same subtype of ovarian cancer or any type of cancer, that really makes it so difficult to treat this disease in general. And that's why I also believe that in the future, we have to shift the way how we diagnose and treat patients. And the new approach that I propagate for that and many others is called precision oncology, where we do take a very comprehensive view of these tumors and based on that, for every patient, personalize the treatment that is provided.
I was working in my master thesis here in ETH actually, and I came across Bernd's group and looked into all the research. And I think you spend a lot of time in academia working in pure science and trying to look at new phenomena, trying to understand mechanisms. And for me, and I think for a lot of people, there's this frustration in trying to translate those findings into the clinics. And it's this concept of the bench to bedside gap, right? That it's very difficult to apply what we have in the labs directly to that patient. And I think here, working with Bernd, we're sort of in one of those very lucky, fortunate places where we can start to really see our findings translated. And for me, that's why I wanted to start this project, and that's why I wanted to get involved, I think.
And originally, how this project or the general topic emerged in my group is that we are part of a large consortium that is called the Tumor Profiler Center. And that is a very close collaboration between oncologists, pathologists, so many clinicians, as well as research groups with different expertises. And in this project, tumor samples are taken from a patient. These samples are then sent to the different research groups. These analyze this tumor sample very comprehensively. And in the end, a report is written that then goes back to the molecular tumor board that can consider these data actually to change or finally do a personalized treatment decision for a given patient. And in this context, we also analyze ovarian cancer patients in collaboration with Viola Heinzelmann at University Hospital of Basel. And in the first phase of this project, we saw really interesting findings, and that really triggered then the interest to really go into trying to better understand what are these cell populations that are actually resistant to chemotherapy. And in addition, Viola Heinzelmann, I mentioned before, it's very rare that such samples are actually taken, but she, over her whole career, collected these samples first in Zurich, then in Australia, then in Basel, and so James has now this unique resource for his research available. Every research project really lives from having highly motivated, brilliant young students that work on the project. And in this case, there's James that is really driving this project forward, spending also a lot of his free time on the project. And ultimately, we need to fund these students. And unfortunately, often, if a project is highly interdisciplinary, in this case between basic research and clinical research, it often doesn't fall under the standard funding schemes that are available. And in such cases, getting support and funding for young researchers like James from a foundation is really essential for us.
Well, personally, it would be to find out what is happening in all of these patients. And I think we're starting to see, and as Bernd has continually mentioned, that there is this huge heterogeneity in them. So finding multiple resistant mechanisms and multiple ways that these cancers can come back would be extremely rewarding, I think. And then because we have this quite fantastic new technologies to look into these tumors, to be able to then go into these resistant parts of a tumor that we've then identified and potentially look at what else, how they're behaving, what else they're doing, and to try and predict what other drug might be suitable for this patient. And to see this sort of decision being eventually included into the clinics would be really rewarding, for sure.